Good evening, everybody. Welcome to Library Hall. And tonight is, I got it down for Dawn. I'm not tonight is kind of the culmination of what's ended up being a 3-program run of informational programs about Alzheimer's. So Dawn has been appearing in the audience with our local neurologist and films that we've done about Alzheimer's over the winter and spring, and we're really excited to hear from him finally. He's been like our best audience participant for all of these things, so we're really excited to have him be front and center stage here. Beyond that, I just want to give you a couple heads up of things that are going on right now.
We need
as
more
we're coming
chairs.
up on a deadline,
What?
and I
We
will
need
come
more
back and get you
chairs.
guys more
Yeah,
chairs
good.
as soon as I finish this. The deadline, if you have any young people in your life, children, grandchildren, neighbors, who are 40 and under, our literary festival is celebrating its 32nd year this year. It is one of our biggest signature events with the Werner Library. It's happening on September 6th this year, and the library is scholarshiping 20 people for literary sojourn once again, and I'm still taking nominations for one more week. So if you know somebody who's a book lover who might want an incredible day at our author festival this year, somebody who hasn't experienced sojourn before, and really wants that opportunity to get their foot in the door. It's a $75 ticket. We have raised our ticket prices in over 25 years, which is something of a feat in itself, but we sure know how hard it is in this town to be young and being a teacher or a nurse or something and get your feet on the ground here. So please let me know if you have any nominations. You seem like a good crowd for that kind of thing. And beyond that, I have one, with a captive audience, one program next week. I just want to raise your alert for next Thursday night. We are hosting a screening of a brand new documentary film that does not have distribution yet. It is still in the film festivals. It's called Half-Life of Memory. It is about Colorado's nuclear legacy, specifically Rocky Flats, where they made the triggers for nuclear bombs. And that site had a series of accidents over the years, fires and whatnot. It's been effectively, dirt has been put over things. It's had a level of cleanup that there's quite a bit of discrepancy on how good that cleanup is. And it is now a national wildlife refuge where you can go and walk your dogs and run all over it. So the filmmaker will be here with the film next Thursday. John Lipsky, who was the lead FBI investigator on the accidents at Rocky Flats, will be here, as well as Judy Pitia, who's a veteran worker from Rocky Flats for decades and has now become kind of an activist on that piece of property in Colorado's history. And really, they're all kind of, this is in effect to not erase this piece of Colorado history. And so that's next Thursday night, 7 o'clock, right here, same time, same place. With that, Dr. John Moss has his own introducer, so I'm going to turn over the mic to John. All right, come on.
Thank you.
Hello, I'm John Norton. I'm the executive director of a corporation that we put together to take this drug that you're going to come hear about through the FDA process. And if you're at all worried, better people will follow. Okay. So welcome. I'm Don Moss, received his bachelor's and PhD degrees from Colorado State. Now Dr. Moss, and then Dr. Moss, he completed a 2-year Sloan Foundation Fellowship in Neuroscience at the University of Chicago, San Diego, where the seeds of this 40-year legacy were planted. 40 years. He accepted a faculty position at the University of Texas, El Paso, where he's now a professor emeritus. During that time, he's published more than 100 scientific articles and notes in respected peer-reviewed publications, book chapters, and has been featured as a speaker in conferences around the world. In almost every case, these publications were about the discovery and definition of a new, may I say, revolutionary treatment of Alzheimer's and associated disease. I am so enthusiastic about this work that Don made me make a promise that I would restrain this introduction. Therefore, I can only add that you have a treasure of living in Steamboat and buried treasure that he'll talk about. Ladies and gentlemen, please welcome my friend and your name, Dr. John.
Good job, John. Well, thank you to the library and Jenny and John and everyone. Thank you all for coming out tonight, spending a Thursday night to hear about Alzheimer's disease. And Alzheimer's disease is a very serious problem. And as John alluded, I started working on Alzheimer's disease in about 1982, and I got my PhD in 1973, so that's 50-some years ago, but I've continued to learn since I got my PhD that long time ago. But it's really special to be here tonight with all of you, my friends, and people that I will become friends with. Is this mic working okay? Okay, but what I'm gonna do today is I want to approach Alzheimer's disease from the disease standpoint. I'm gonna try to tell you everything, I will tell you everything I know about Alzheimer's disease, but there's a lot to know. I won't be able to tell you everything. And my objective tonight is that nobody will go away from this meeting tonight with a question about Alzheimer's disease that you failed to ask me. And it doesn't make any difference whether I brought it up in my talk or not. If you have a question about it, ask me. I may not know the answer, but we'll see. We'll figure it out. A lot of what I'm going to talk about tonight is my frustration with Alzheimer's disease, as is the lead line up here on my first slide. And I expect nobody in this audience is satisfied with where we are on Alzheimer's disease. Is anybody in here satisfied with Alzheimer's? The progress that we have not made since, Dr. Alzheimer discovered it and named it in 1907. We haven't been working on it since then, but in 1976, the National Institute on Aging was established, and they made Alzheimer's a priority, and we have made essentially that's too harsh. We haven't made enough progress by any means. So I'm going to talk about what goes wrong in the brain of Alzheimer's patients. I'm going to talk about why it always winds up in dementia, the severe memory loss. I'm going to tell you what we need to do about it, and we know very well what to do about it. And I'll tell you why we're not doing it, and the drugs that we have are essentially useless. Have I forgot anything? Oh yeah, and what we're going to do to avoid Alzheimer's at the end. Don't let me forget to talk about that. It's so embarrassing when I forget what I'm doing, you know. My students used to tell me if I ever figure out anything to write it down. Okay. So anyway, yes, I'm a professor emeritus. Most of or a lot of what I'm going to talk about, not everything, but a lot of what I'm going to talk about relates to two things: what Alzheimer's is as a disease and what we need to do to get a better treatment. Those are the two main things. And I don't give medical advice, and it's probably a very good thing that I don't give medical advice.
You know why? Because you've got 2 mics.
Oh, okay. I'll just use one mic. Is that okay? Then I won't have to repeat myself, right? Okay. Alzheimer's disease was named by Alois Alzheimer in 1907. Oskar Fischer also described 12 patients the same year. Dr. Alzheimer described one, Auguste Dieter, that you see here. It was named after him. But anyway, that's just a historical note. Dr. Alzheimer described Alzheimer's disease on the basis of 2 features that he saw in the brains of postmortem patients. One of these, can you see this pointer at all? Yes. Okay, one of these is the senile plaque. You see a senile plaque, senile plaque, senile plaque. And these senile plaques showed up in 1907, and Dr. Alzheimer's did not know what was in the plaque, but they were in the brains of Alzheimer's patients. We now know that they contain an abnormal protein, beta amyloid. Now, I call it an abnormal protein. Beta amyloid is a normal constituent of the immune system in the brain. It's an antimicrobial protein, and the reason we have beta amyloid in large amounts in the brain is because the real cause of Alzheimer's disease, in my opinion, and not everybody agrees with me, but it's inflammation in the brain. And this is one marker for inflammation. Another one over here are the neurofibrillary tangles. Dr. Alzheimer saw those. He has these stained so that he could see them in his microscope. And we now know that these are made up largely of a hyperphosphorylated tau. And that's a protein. Tau is a protein that is a normal protein in the brain, and it is essential for the function of the neurons in the brain. It's a microtubule associated protein, but when it gets hyperphosphorylated, it quits functioning properly and it results in some brain failure. We now know that these 2 things are tombstone markers, and I call them tombstone markers because they are what you see at the end of life in Alzheimer's patients, where these processes have been going on for 15, 20 years before you pass away and your brain would go to autopsy and they would be seen. Earlier in the disease, before these abnormal proteins are formed and aggregate into these bricks in the brain that shouldn't be there, these are soluble proteins, and they are participating in the biochemistry of the brain, and they're screwing up the biochemistry of the brain. This form of these proteins is probably not all that toxic because they're sequestered and they're walled off from the rest of the brain. But when these proteins are soluble in the neurons, they're toxic. Now, the reason that Alzheimer's disease always ends in dementia is because a third thing that was discovered about Alzheimer's disease, and was not discovered until 1976, when Dr. Peter Davies and Maloney discovered that the brain of Alzheimer's patients are missing an essential neurotransmitter. And a neurotransmitter is a chemical that's released by one neuron and has to communicate to the next neuron. It has to be received by that neuron. It has to stimulate a change in that neuron, and it responds to it so that the brain functions. The neurons communicate with one another, and you get these really complicated, actually elegant functions in the brain are because of these chemical messengers called neurotransmitters. The essential neurotransmitter that's missing in Alzheimer's disease is acetylcholine. This is also called acetylcholine. If you're a chemist, you would call it acetylcholine because that's what it is. It's acetylcholine. But if you're a pharmacologist or a medical doctor, we call it acetylcholine, and I hope that's not confusing. But if you don't have acetylcholine, you don't have memory. And so what happens in Alzheimer's disease is that the basal forebrain here, the basal forebrain cholinergic system, this spot right here, disappears in Alzheimer's disease. Now, this is not to scale. This is not what the basal forebrain looks like in your brain. The basal forebrain is about the size of a small marble on the base of your brain here, but its connections are essential. The medial septal nucleus here is connected to the hippocampus. It sounds like, a song. The septal nucleus to connect to the hippocampus. The vertical nucleus of the diagonal band of Broca and the horizontal nucleus of the diagonal band of Broca then are connected through, and the nucleus basalis minor are connected to the cortex. Now, I have to apologize. It has been mentioned in my presence that sometimes I outgeek myself and I go on and on about stuff. And some of it used to show up on class evaluations. But anyway, this is the problem. The basal forebrain cholinergic system disappears. And when that disappears, your memory disappears. And that's the origin of dementia, is right there. Now, the question that we have to deal with is if you don't have enough, and I'll tell you, I'm going to focus on what we have to do about dementia. And at the end, I'll tell you what we maybe have to do about Alzheimer's disease in general. I'm not going to talk about inflammation. I am not going to talk about beta amyloid. I'm going to talk a little bit about hyperphosphorylated tau, but I'm going to focus on what we have to do to rehabilitate people's memory with drugs so that you can have your life back, so that you don't go to nursing homes, so that if you had Alzheimer's disease and it was treated well, you might not even find it all that inconvenient. I'll tell you, we can do a lot better than we're doing right now. In fact, I'd like to say, how many people in here know about Parkinson's disease? Everybody knows about Parkinson's disease. And you know that there's a treatment for Parkinson's disease for the symptoms. Very good at controlling symptoms. It's called L-DOPA. And when you take L-DOPA, well, Parkinson's is a movement disorder where you have trouble controlling speed and coordination. You can't move. Parkinson's is a bad disease. Alzheimer's and Parkinson's are the two major neurological diseases that we have to contend with. But the point I'm going to make here is that if you take L-DOPA, what happens? You get better. You can tell if you're on L-DOPA. You begin to walk around. You begin to get normal coordination. When you take the drugs for Alzheimer's disease, the point I'm going to make is, can you tell whether you're on them or not? No. But anyway, I'm off on a track here. So what we have to do is we have to replace acetylcholine so that when you get a drug to replace acetylcholine, you notice a reduction in your dementia. And people around you notice that you have a reduced dementia. Okay, how how to, I didn't proofread my slide, did I? How do you replace acetylcholine? I just mentioned with Parkinson's disease, I can give you L-DOPA. L-DOPA goes into the brain, it gets converted automatically to dopamine and you get better. We can't do that with acetylcholine because the precursors for acetylcholine, the metabolism of acetylcholine just doesn't work that way. You can't just give you a precursor and it works. So how do we replace the acetylcholine that's lost in the brain to treat the dementia? And the answer is that we can't do it the other way. So what we have to do is we have to give an inhibitor for acetylcholinesterase. And acetylcholinesterase is the enzyme that breaks down acetylcholine. And if we stop it from breaking down acetylcholine, then the first thing you know, it builds up, it amplifies the effect of acetylcholine. It works like an amplifier, it enhances acetylcholine. And that's the only strategy that we know of that works. And with your permission, instead of calling it acetylcholinesterase all the time, I would like to call it ACHE. Would that be okay? And I may slip and call it acetylcholinesterase once in a while. But in 1993, actually there was one drug approved for Alzheimer's disease before 1993. It was called Tacrin, and it was sold under a brand name of Cognex, and it was really bad. And it caused liver problems and everything. But donepezil, this one here, was the first fairly successful acetylcholinesterase inhibitor, ACHE inhibitor, to treat Alzheimer's dementia, and it was introduced in 1993. 1993 was 32 years ago, if my math is correct. And then there were two more that were approved, rivastigmine, which was sold as Exelon, and galantamine, which was sold as Rimonel, and they up through 2001. These are the only drugs that are available today to treat dementia. There is one other one called memantine, but it works by a different mechanism. It's not an ACHE inhibitor, it's a calcium channel blocker trying to stop the toxicity in the brain from calcium. But anyway, none of them work. Well, they do work a little bit. I will tell you how well they work, and you make up your mind. Like I've already said, L-DOPA works for Parkinson's. That's what we need now. Has anybody seen the movie Awakenings? It's an old movie, but it's the true story of, I think it was Oliver Sacks that came up with L-DOPA and how it worked. People they worked with people that were catatonic. They had an inability to move at all. And they gave them L-DOPA, and within a day they were up walking around. That's what we want for Alzheimer's disease. We don't have it yet, and that's one of the reasons that I'm frustrated. Okay, so why don't our drugs give immediate release from dementia? And the reason is that the drugs we have were poorly designed. And I'm going to go into I'm going to get a little geeky about drug design, but it's how to avoid the whole problem. The whole problem is that ACHE inhibitors and acetylcholine works everywhere in the body. Not everywhere. I mean, there are other systems, but acetylcholine works in the brain for memory. Acetylcholine works in the brain to control sleep. Acetylcholine works in all of your autonomic ganglia, your sympathetic and parasympathetic ganglia, to control all the smooth muscle, cardiac muscle, and glands in your body. Acetylcholine is running my skeletal muscles right now. And so if I'm going to give you a drug that starts messing around with acetylcholine, if I'm going to start giving you a drug that will greatly amplify acetylcholine, then the first thing you know, you have a huge problem with toxicity. And the toxicity is that the intestines, it's a real low-tech problem. Your intestines run on acetylcholine, and they are very sensitive to drugs that enhance acetylcholine, and so you wind up with nausea, vomiting, and diarrhea. So the reason I can't treat dementia is because the patients don't like having nausea, vomiting, and diarrhea all the time, and the drugs are intolerable, and so you can't take the drug in an adequate dose. It's like being diabetic and you only get a third of the dose of insulin. It won't work. And it's been that way for 30 years of not working. It's been the same problem for 30 years. It's not new. The problem is, as I mentioned, donepezil has been measured 3 times in Alzheimer's patients that are being treated with donepezil, and with some really modern techniques now, you can take and you can give people a positron emitter while they're taking the drug, and they're walking around, and you can tell what the inhibition is in the brain. You can tell how effective it is in the brain. It's a lot better than in the old days where you'd have to take a biopsy. Same thing with rivastigmine. Same thing with galantamine. All 3 of these drugs are ineffective because in one study they found it was 27% inhibition in the brain, 32% inhibition in the brain, 19%, 31%, and 38% inhibition in the brain of patients. We need a minimum of 50% inhibition to be effective. We're not even close. They do work a little bit. They're better than nothing at all. I'll talk about that more later. They're totally inadequate. The lack of selectivity of the drug for the brain is the whole problem. If I had an acetylcholinesterase inhibitor, an ACHE inhibitor, that would work effectively in the brain and not work in the rest of the body, I would be able to treat dementia. Now, I don't know, does anybody in here have myasthenia gravis? Oh, good. It's never good to know what I'm talking about. But anyway, it's a disorder that's also treated by ACHE inhibitors, but it's a problem in the peripheral muscles. It's really easy to have an ACHE inhibitor that works in the rest of the body and not the brain. All you have to do is make it a quaternary ammonium, put a little charge on it, and it won't go into the brain. And it's used in myasthenia gravis, very effective. The opposite is not easy. To get something that'll work in the brain and not in the rest of the body is almost impossible because of the blood-brain barrier. And this is what the problem I had to work on. We needed a new brain-selective acetylcholinesterase inhibitor. This is, I want to point out, this has been a barrier for 30 years. There are 500,000 new cases of Alzheimer's disease every year. There haven't been that many 30 years ago. There weren't quite that many, but let's just round off. Give me a break on some numbers here. But 500,000 patients a year that have not received adequate treatment for 30 years is 15 million people have not had appropriate treatment. And it's not that difficult. So we need a new brain-selective ACHE inhibitor. I need something that works in the brain and not in the rest of the body. And so that's the problem I worked on for a while. Now, again, I'm going to out-geek myself a little bit, and just hang in here with me. This is a schematic of the catalytic site of acetylcholinesterase. This is ACHE. These represent protein strands here, amino acid strands. Acetylcholinesterase is a protein that has a molecular weight of about 260,000 or so. It's a big protein. And the ACHE inhibitor that I'm going to talk about is methane sulfonyl fluoride, or MSF. And MSF has a molecular weight of a little less than 100. It's a little tiny molecule that gets right in the catalytic cleft of the enzyme where acetylcholinesterase breaks down acetylcholine. Acetylcholine has to fit in there, and acetylcholinesterase breaks it apart. MSF fits down in there, reacts with the enzyme right there, and then that kills that protein. It doesn't just plug up acetylcholinesterase so that it's not very efficient. It will kill that protein molecule. And that's important because, well, and it does that all over the body. It kills acetylcholinesterase everywhere in the body that it goes to and gets to. But what's important here, and I discovered this was an experiment in my lab in 1985 or so, that acetylcholinesterase in the brain is replaced at a very slow rate. So if you go in and you target that in the brain, it lasts and lasts and lasts. But in the intestines, it's replaced very quickly. This is the first time we've had an opportunity to really separate acetylcholinesterase in the brain from the intestines, and that has to do with how fast the body replaces them. You know, all of the proteins in your body are replaced. They turn over in a regular cycle. Some of them are faster than others. In the intestines, acetylcholinesterase is replaced in the intestines with a half time of 1 day. So if I give you MSF, I'm going to talk about this drug, it's an irreversible inhibitor. I give it to you in the intestine, and I want to talk about this. This Y here is inhibition, so I'm going to be talking about how much inhibition accumulates in the tissue, but I give you a dose of MSF that produces 10% inhibition. It will produce 10% inhibition in the intestine, and it'll produce 10% inhibition in the brain. No advantage there yet. But I wait a day, and then I give you another dose of MSF. And then I wait another day and give you another dose of MSF and another dose. And this represents 21 days of giving both rats and humans. This is representing what happens in both rats and humans and monkeys, too. I've done experiments in monkeys. The intestines recover so quickly that you never get above about 20% inhibition in the intestines. Your intestines don't even know that you're being treated with the drug. But the brain recovers very slowly. And remember, I told you that donepezil, rivastigmine, and galantamine can't produce more than about 30%. Remember that slide? And they can't produce more than 30% because if you get above 30%, then people start getting nausea, vomiting, and diarrhea, and then they don't want the treatment anymore. Well, look here with MSF, we expect to shoot right through 30% in the brain, 40, 50, 60, even, and I'll show you where we get into 70, and I've had monkeys up to 80 and 90% inhibition in the brain, and they don't lose weight, they don't quit eating, you can tell when they have diarrhea and vomit. You can't tell when they're nauseous except when they quit eating. This is what is supposed to happen with MSF because it's an irreversible inhibitor. It's the only inhibitor that is an irreversible inhibitor. So this is really great, you know, if you're going to do these calculations and you have all these expectations, what in fact happens? Well, this is a lucky break. For the first time in history, that's a lucky break. The brain is different than the intestines. And you can do this with 10% inhibition each dose, a little tiny dose. And you can avoid a lot of toxicity and it accumulates. For the first time in history, Alzheimer's dementia can be treated without limits from nausea, vomiting, and diarrhea. Now I'll get to the next slide that I forgot about those other two along the way there. But anyway, this is what happens when you actually take a group of rats and you treat them for 21 days, just like I was describing there. And you do it for 21 days, and then at the end of 21 days, then you sacrifice the rats and you remove the really important organs that operate on acetylcholine. You check the intestine, you check the skeletal muscle, you check the heart, and see how much inhibition is there. And I think my calculations suggested they would be about 20% or less. That's real close. That's about as good as you can get between calculations and real world outcomes. But look here, what happened in the brain. The brain of those rats was about 75% inhibited. This was a crowning achievement. This is how we need to treat humans. We can treat dementia if we get over 50%. Everybody remembers that and if you can get to 60 or 70%, then it's even better. So that worked out real well. This effect is unparalleled by any drug in the world. And this goes to my frustration. Remember I told you that I discovered that the brain had a slower ACHE turnover than in the other tissues of the body back in I think it was about 85. Well, that's only 40 years ago. I published the paper. It's been out there. This is not a secret. Did anybody follow up on it? No, I had to do it myself. Okay, I had to do it myself. So those were in rats, and this is in Alzheimer's patients. And this is my only human clinical study that I've run. And I was a professor at the University of Texas at El Paso at the time, and I made some acquaintances with a doctor in Mexico that was very interested in Alzheimer's disease, and we proposed to do a human clinical trial in Mexico because it didn't cost hundreds of thousands or millions of dollars like it did in the United States. And so we did this experiment, and it was approved by the University of Texas at El Paso Institutional Review Board. It was approved by El Departamento de Consumos y Medicamentos in Mexico, and it was approved by La Universidad Autónoma in Chihuahua. We did all the approvals. It was not a renegade experiment out of somebody's garage. It was all done correctly. We recruited these, and these were in Mexico, and we recruited these patients, and I have to explain this for a moment. This was a double-blind, placebo, randomized experiment, which are all the gold standard things. The patients did not know whether they were on the drug or not. The people that were evaluating did not know. They were blind, all of that. But I had to make it a crossover experiment. And I had to make it a crossover experiment for two reasons. One is, in a regular experiment, you might just take 100 people and divide the group in half, and 50% of the people get the drug and 50% of the people get the placebo. But I ran into a wall because none of the people that I wanted to recruit for the experiment wanted to be in the placebo group. Oh, you have a 50%, you're gonna do this for 16 weeks and you don't know whether you're gonna get the drug or not? You know, you're signing up for 4 months of this and all that, and people didn't like it. And I had a lot of trouble getting very many people to do it. And also, if I make it a crossover experiment, I get to double the efficiency of using the patients. In other words, in a crossover experiment, I'm going to start out with, in this case, this was a small study. It was in 15 patients. And if I'd have made it a parallel groups design, a regular one, it would have been 7 in one group and 8 in the other. You know, but when I do a crossover experiment, I take half of the patients and I start them on MSF, and half of the patients start on placebo. And again, they don't know whether they're on the drug or not. And we assign them an arbitrary value of 0. We measured their memory at the start of the experiment, and whatever it was, we said, okay, that's your baseline, and we're going to call that 0. And then they were treated for 8 weeks, and some of them received MSF, and some of them received placebo for 8 weeks. Up means that when we measured their memory 8 weeks into this, they were improved. On the placebo, when we measured their memory 8 weeks later, they had not improved. And if I can, I don't want to out-geek myself here, but this is a crossover experiment, and everybody in it knew that it was a crossover experiment. So I had to tell the patients, you're going to be on the drug part of the time. And I had to do that for ethical reasons. People have to do informed consent and all of that kind of stuff. And I told you that it was placebo-controlled, double-blind. Well, with the old drugs, the ones that are available now, if you run a double-blind experiment with the drugs we have now, guess what? The people that are vomiting and nauseous are on the drug. And the people that are not doing that, you know, may I please be in the placebo group for the rest of this experiment? Well, we had the other problem. We had the opposite problem. We told everybody that they were going to be crossed over and that they might start on placebo, they might start on MSF, and you don't know. Well, the trial unblinded itself because some people started getting better in an Alzheimer's trial. And if you're an Alzheimer's patient, it's a chronic disease, and you don't generally get noticeably better. And I do speak Spanish now, but when I was working in Mexico then, I didn't speak Spanish very well, and I had nothing to do with the evaluation of the patients because all the testing and everything was done in Spanish and stuff. I was the director, but I didn't have anything to do with the patients. And so I had a laboratory in the back of the clinic where I would go back and keep my notebooks and take care of drug making and all that kind of stuff. And one day, a young woman, probably 40, came back to my laboratory in the back of the clinic. And it was really fun to work in Mexico. I loved working in Mexico. It was great. All of the patients had caregivers that knew what was going on and all of that. And to be a doctor down there, a professor doctor down there, they thought that was, it was more respected than it is here, let me say. Anyway, this young woman came back into my laboratory at the back of the clinic, and she was a little bit forward, and she walked up, I had a white coat on, she walked up to me and grabbed my white coat, the front of my white coat, and she said, do not take my mother off of MSF. And that's because we were getting close to Christmas. And her mother was in this group that was getting better and better and better, and we were getting closer and closer to the 8 weeks where she knew that her mother was going to be put on placebo, and she came back and told me not to take her mother off of MSF. They wanted to have a nice Christmas. And I said, gee, you know, I'm sorry I can't help you. It's all set in stone, you know. Thank you for your input, but, you know, whatever, que sera, sera. And I didn't. But anyway, and by the way, after Christmas, the same lady came to me and she said, thank you for not taking my mother off of the drug. And I said, why do you think I took your mother off of the drug? You know, I didn't tell her, I didn't do anything. That would have broken the whole experiment, right? Well, I was making the drug in peanut oil and making it in capsules, and some capsules just had peanut oil and some capsules had peanut oil with MSF in them. And it was Christmas and it was in Chihuahua, Mexico, and the weather was getting cold. And she'd been looking at all those capsules, looking at them really carefully, and they didn't change appearance over Christmas. They were cloudy before going up to Christmas, and they were cloudy after, and she thought I had not changed she thought I'd changed the drug, but I hadn't. When it gets cold, peanut oil gets cloudy, and she was seeing it cloudy and cloudy and cloudy, and she thought her mother had stayed on the drug, but her mother did not. On the other hand, then the other group got worse, and then when we switched over here, then they took MSF and they got better. And they got better so that you could notice it, because one of my assistants that was out with the patients all the time came back and told me that the patients were talking among themselves. Is your patient on the drug now, or are they in this group or that group? It was noticeable. It was very nice. But anyway, at the end of this, then at the end of 16 weeks, we gave them all a test, and they were all better than when they started, which was a surprise to me, and I'll talk about that later. Because the people that got better on MSF, when we stopped giving it to them for 8 more weeks, you know, what's going to happen when you quit taking your pills for 8 weeks? You know, you're supposed to get worse. And you see up there that they didn't. The placebo, it's this group right here that I'm talking about. They should have crashed. They should have come down and been down at this level, but they weren't. And I'll talk about that. Okay, now, so I've been bad-mouthing donepezil and rivastigmine and galantamine, but this is a plot, a comparison of the results that I got with MSF on the same graph using the same tests as others have done with donepezil and rivastigmine. These come from the data inserts of these drugs. The blue is donepezil, and donepezil has produced about 1, maybe 2 points of improvement when the patients were on donepezil, and the same with the rivastigmine. It was about 1 point improvement on this measurement of memory over here. And compare that with MSF that I showed you in the prior slide here. There were 6 points improvement here over 8 weeks of MSF treatment compared to 1 or 2 points improvement on the other one. And then I've circled this up here. That's because in that experiment with these patients, I had 66% inhibition in the brains of those patients. And that's phenomenally better than what you get with 25 to 35% inhibition with donepezil and rivastigmine. That's the difference between an effective and an ineffective drug. Now, I want to go back to the people that started out on MSF and then they didn't get worse for 8 weeks when they were off of MSF. The question is, what was happening there? Why does memory enhancement last longer than the direct effect of the drug? This experiment was published in 1999, and the experiment was done in 95, and I didn't get it published until 99 for various reasons related to patents and stuff like that. But anyway, in 1999, I did not realize how important that was, that the patients did not crash when they went off of the drug. They maintained that. Now, this is not my work, this is some work that others have done that is revolutionary in Alzheimer's disease. These experiments were done by some people in Paris, some really excellent people in Paris, Bruno Dubois and Erica Cavedo and others, and they published it in 2015. Okay, now 2015 was 10 years ago, right? And have you ever heard about this being a breakthrough in Alzheimer's disease in 10 years? No, I haven't either, and therefore I'm frustrated again. I'm easily annoyed. I should have told you that. But anyway, what Dubois and Cavedo and their colleagues did in Paris was a really wonderful experiment. And I don't know how they got it done, but they probably had, I think it was 250 or 300 patients that are enrolled in a memory institute in Paris, and these are prodromal patients. Now this is very important because a prodromal patient is somebody that has all of the red flags for being an Alzheimer's patient, and they are beginning to have memory problems, but they are not yet diagnosed with Alzheimer's. They are prodromal. And the reason I'm emphasizing this is that if we're ever going to avoid Alzheimer's disease, we've got to start treating them before they show up with dementia. Now, we can improve their memory, as I showed you in the experiment that I did. We can improve their memory after they have dementia, but that's not really the productive way to treat or avoid Alzheimer's disease. You don't want to wait until you have dementia and then try to repair the damage in the brain with a drug. So anyway, they did it in prodromal patients. They took, let's just say, I don't remember the number, let's just say, make it easy on me. They had 300 patients and they divided them 150, maybe it was less than that, but anyway, they divided them in half. And one half got placebo and one half got donepezil. And donepezil is a drug that's not effective at treating dementia very much. Remember, it's pretty mediocre. But they did this for a year, and at the end, they did the experiment where they did a detailed MRI on the patient's brains. At the beginning of this, they went a year. One half got the drug, one half did not get the drug, and at the end of a year, then they did an MRI on all of the brains of the patients again, and they found that donepezil, an ACHE inhibitor that is not supposed to do anything but treat symptoms, they're supposed to make your memory better on a day-to-day basis, were slowing the annual rate of hippocampal atrophy. Remember I said one of the brain areas that's important is the hippocampus. It slowed the atrophy of the basal forebrain, the origin of acetylcholine that is disappearing. It's disintegrating and disappearing But donepezil slowed that down. And it also slowed the thinning of the cortex. This is a breakthrough because I'm sure that you've heard that we've had these new drugs approved for Alzheimer's disease that attack beta amyloid. The brand new one, there are 3 of them that were approved for a different purpose, to slow down the disease. The people who worked on those drugs would be dancing on the board of directors table if they got results like this. And one of those, anibibabab, it's a monoclonal antibody that was developed to attack beta amyloid. It was a big deal about 3 years ago, 4 years ago. You could have that for $57,000 a year, and 30% of the patients had swelling in the brain and life-threatening swelling in the brain and all these other side effects, and they're still working on beta amyloid, which, anyway, look at this. The French did this, and nobody pays any attention to it. By the way, donepezil costs about $3 a dose. It's a generic, and that's what they were using. Well, you'll never get a big drug company to run a trial on that because it's a generic and you're screwed. Okay, so what I'm proposing, looking forward, is to make some changes about Alzheimer's disease treatment. First of all, the Alzheimer's Association hired an expert to look at the population of patients and come to a conclusion. Okay, what if we could get a drug that would delay the onset of dementia? In other words, about 20% of us are going to have Alzheimer's dementia. I hate to tell you that. You know, we can count off 1, 2, 3, 4, 5, and 20% of us are going to, we want somebody else to get the other. Well, you know, and it gets worse as you get older. You have about a 15, 20% chance of having dementia by the time you're about 65. By the time you're 75, you have about a 25 to 30% chance. By the time you're 85, you have about a 50% chance of having dementia. Well, I just turned 80. I'm worried about this too. But anyway, what if you had a drug that would delay that? Instead of a person who was going to get dementia at 70, what if you could get them delayed? What if you could give them something to delay it until 75? Or people that are going to get it at 65, they can delay it until they're 70. If you could delay the onset in the population in general 5 years, you could reduce the estimated 13.5 million people that we're going to have with AD, Alzheimer's disease, in 2050 by about half. We could do away with about half of the dementia if we could just delay it. Why? You're old and you're going to, unfortunately, pass away from something else first. But you're going to pass away with something else first anyway. You might as well live well until you do it. Or then, this is one hypothetical drug. They said, if you had a drug that would do that, how much would it help? And the answer is, if you had a drug that delayed the onset, it would help a lot. Then they said to this guy, they said, okay, what if we didn't change the delay of the age of onset? Let's say everybody gets it anyway, but instead of the 5.5 years that you normally have between the time that you get diagnosed with Alzheimer's disease until you pass away, instead of that 5.5 years being spent, the last 3 years of it in a nursing home, what if we could slow the progression of the dementia where you have memory problems, but they don't get very bad. they get bad slowly and you have a better quality of life and you don't require as much care. They didn't say how much it had to slow it in that study, but it would reduce the patients with severe dementia by 80%. And the severe dementia is the really bitter, awful end of Alzheimer's disease. You could get rid of most of that if you could slow the progression of the dementia. You could slow the people with severe dementia from 6.5 million down to 1.2 million in 2050. So this is a good strategy. It makes it well worthwhile, okay? ACHE inhibitors are not going to perhaps keep you from getting Alzheimer's disease. And in fact, I have to emphasize that the development of Alzheimer's disease in the brain precedes the onset of dementia by 15 or 20 years. And so if you're 55 or older in this room, you may have Alzheimer's disease and it won't result in the onset of dementia for another 15 or 20 years. But we could get a lot of relief if we could get that to do that. Now, what are we going to do about these almost useless drugs? We must do better. This is where I really work on it a lot. What we need to do is we need to get a better ACHE inhibitor that works selectively in the brain. There is only one ACHE inhibitor in the world that works selectively in the brain, and that's MSF. And it's not on the market. And I could go on and on a whole night telling you why it's not on the market. It's because of patent problems and money, big pharma, and one thing and another, but it's a great drug. So, and it's not the only one that could be developed. There could be another one that's developed if anybody used the same strategy that I did. And I've been telling them about it since 1985 and nobody's done it, so I don't think we're in any danger there. They're not going to start now. But the other thing we can do, let's say we do have a better ACHE inhibitor, that would be great. But even if we don't, we need to start ACHE inhibitor therapy before you get demented. Full disclosure, I read those articles on the French study in 2015 about donepezil slowing the atrophy of the brain. And I've been on donepezil since 2016 as a prophylactic treatment. It's like compounding money in the bank. So you get 2% a year or something. Well, over 10 years, 20 years, it makes a big difference. And I would propose that we make a very simple change in the way we treat Alzheimer's disease, and that is that we start patients on ACHE inhibitors as prophylactic patients. Now, it will never be approved by the FDA. Use will never be approved. Why? The FDA requires what is called an adequate and well-controlled experiment, and you have to get data. Well, I could do an experiment for prophylactic use, but that would mean that I would have to go out and I'd have to get 100,000 patients that are 55 years old and follow them for 30 years, and half of them would have to be on donepezil and half of them would have to be on placebo. And I don't know, that'd be $1 billion. And these drugs are generics. And so you get all done, and the drug company and their stockholders would be left holding the bag for the cost of the experiment. And a generic drug company could produce it and sell it to the patients for 50 cents a dose. It's just never going to happen. But it would be pretty easy. I had to go to my physician and He knew I worked on it, and this was a long time ago in 2016, he said, well, if you want to try it, go ahead, I'll write a prescription. You have to get a prescription for it. And so he did, and so I've been taking it, and then in the meantime, I've changed doctors a couple of times and moved and stuff, and every time my doctor will go along with it. These drugs are safe. Millions and millions of people have taken them for years and years. Once you start taking donepezil after you have dementia, you're going to be on it 5 or 6 years, or if you're lucky, longer, or maybe not. But there's no risk, there's no downside to it, and it could make a big difference. If somebody is 60 years old and has, let's say, a genetic predisposition for Alzheimer's disease. Let's say you have ApoE4, gene for ApoE4. It's an allele. And you get one from your mother and one from your father, one of these alleles. And there are other forms of it. There's ApoE2, ApoE3, and ApoE4. ApoE4 is not all that common, but if you get it, if you get one copy from your mother or father, your risk goes up. If you get 2 copies, one from your mother and one from your father, then your risk goes up even more. You'd be a great candidate for it. If you have a family history of Alzheimer's disease or you're worried about it, you'd be a great candidate for it. But then again, like I said in the beginning of this, nothing I tell you here is medical advice. Don't listen to me at all. But this is what I would do if I were in charge. And I don't happen to be in charge. The other thing that we need to do, and I talked about it in the beginning, the beta amyloid and the hyperphosphorylated tau that make up the senile plaque and the neurofibrillary tangles in the brain are the tombstones of long-time inflammation in the brain, chronic inflammation in the brain. And what we need to do is we need to prevent inflammation in the brain. But that's not going to happen in our lifetime. Inflammation is the body's response to injury or infection, and you start messing around with inflammation, blocking inflammation, you're going to get all kinds of side effects, and it's another one of those experiments where it would take 20 years, and they don't even have the drug to do it yet. It'll be 50 years, even with a good run, it'll be 30 years before we get it to address the underlying problem. And they're probably cutting the funding for this. And by the way, can I digress for just a moment? Not that it would make any difference, but I said inflammation is the result of injury or infection in the brain. Some of the big infections in the brain that are associated with Alzheimer's disease are from herpes viruses, herpes type 1 that gives you the cold sores, and the herpes zoster that give you shingles. And there was a study that just came out recently that indicated that one of the benefits of getting your shingles injections is that it reduces your risk of Alzheimer's disease. A big benefit from getting your shingles vaccination is that you don't get damn shingles either. But it reduces your risk of dementia. Okay, again, special thanks to the Werner Library. If you want more information, see John in the back. And this isn't the, okay. I had a slide that says, do you have questions? I said in the beginning of this, my objective is I want to answer any question that you have about Alzheimer's disease if I know the answer. Whether it relates to what I've talked about tonight or not, whether it relates to my drug or not. I know a lot of trivia about Alzheimer's disease. If you have a question, then now is the time to ask it. Yes?
How can we get the University of Texas, El Paso to release the patent on MSF?
Is it possible to get the University of Texas at El Paso to release the patent on MSF? Yes. I applied for the patent for MSF to treat Alzheimer's disease in 1996. And back then, you got 20 years of protection from the day you applied for the patent. Now it's 17 years from the day it issues, but back then it was 20 years from then. So all of my patents for Alzheimer's disease expired in 2016. You may wonder what the hell happened with my patents for 20 years, right? Well, I was an employee of the university, and the university then decided to exert their interest, is what they called it, in my patent, which means that they own the rights to it. It's my patent, my name is on the patent, but they own the rights to it. As my employer, then they got to manage it. They got to license it and all that, and they screwed it up. They frittered it away. It was licensed to 3 different companies during that 20 years. 2 of them went broke. The third one licensed the patent in 2008, when there was only 8 years left on the patent. They ran a wonderful clinical study on Alzheimer's disease in Germany. It was a phase 1 study. It was on normal patients, testing MSF in normal patients, and I didn't talk about that tonight. They gave it a real go, but we didn't have enough patent life left. Nobody else wanted to license a patent that would only have 5 or 6 years left on it at that point, and they still had to run some more clinical testing. And so you get stuck in what's called patent purgatory, where somebody else owns the right to it. It's not long enough to do anything with it, and so you just have to wait and wait and wait. And so the patents have expired and they cannot be renewed. But we are going to pursue FDA approval without a patent. It is possible under what is called new chemical entity regulations, the FDA regulations, and they have similar regulations in Europe and other places. And that is, if you can find somebody that will foot the bill for the clinical studies to get it approved, and I may be too optimistic here, but I think we can do it for only $100 million. If we were starting out without a patent that has not been in humans before, see, MSF has been through 3 clinical trials, it's been in humans, it would probably be a minimum of $500 million, and a lot of drugs run $1 billion. But because we've got a leg up on it, we think we can only do it for $100 million. If we can get somebody to put up the money for $100 million, and I asked my long-suffering spouse over here about putting up $100 million, and she said no, she wouldn't go for that. But if we run the clinical trials that meet the standards for approval, regulatory approval, we get 5 years of market protection in the United States and 10 years in Europe, and that's enough to recover $100 million. I'll tell you how we'll recover $100 million. Am I getting too far out in the weeds here? There are 6 million people in the United States that are diagnosed with Alzheimer's disease today. MSF is so cheap. How cheap is it? You can make the MSF you need for 1 pill for about 2 cents. By the time you make the drug, you formulate it into a pill, you package it in a blister pack, and you distribute it, you could sell it for $2 a dose. If you sold it for $2 a dose, you'd probably be making $1 a dose profit. Let's say you have to sell it for $3 a dose to make $1 a dose profit. Remember I told you that the Donepezil is a generic and you can buy it for $3 a dose right now? That's Donepezil is a generic. MSF could be offered for $3 a dose and you could make $1 a dose. If you could only get market penetration, I'm going to say 50% because it makes it easy to do the math, but it gets easier after that. If I had 3 million people taking MSF every day and I was making $1 a day a dose, I'd be making $3 million a day. I could make $100 million in 30 days. Let's say I only got 20% market penetration, and then I'd have to wait a while. So it might take me 6 months or a year to recover the money for it. And I don't want MSF to be expensive. I think MSF ought to be available to people for whatever it costs to get it approved and make the drug. I think the pharmaceuticals are out of control. But it is very expensive to do the drug trials, and so they have to charge a lot.
Can you get MSF in Mexico or another country?
No, MSF is not available anywhere as a pharmaceutical. The reason it's so cheap is that MSF is used as an industrial chemical, but it's not very pure. I wouldn't take it without it being purified. MSF is a liquid at room temperature. You could simply distill it and have it pure. Who had a question? Yes?
I'm just wondering about genetic testing for the genes for Alzheimer's. I mean, is that possible?
Yes, it is. Now I'm out on thin ice because I don't see patients. I don't order tests or do any of that, but I know they're available. And we happen to have Dr. McCauley right here, who works on Alzheimer's disease, and he works on the genetic variations in people, looking at those that have the genes that allow them to detoxify environmental toxins and contaminants that might go directly to inflammation in the brain. But he knows about all of it. Can you say something about that, Mark?
Well, to answer that question about genetic testing, yeah, you just go to the lab and ask for it. It's simple.
Right.
I had it done and you can do it. Costs, I don't know what it costs, $50, $100. It's not expensive.
Can I ask a second question?
Yes.
The studies that have been done about lifestyle changes, and what's your thought about the benefits of implementing lifestyle changes to prevent onset?
Okay, yes, lifestyle changes are important. Not specifically for Alzheimer's disease, but for brain health, and that's directly related to Alzheimer's disease. Avoid chronic repeated trauma to the brain. That is one lifestyle change right there. And to tie it in with what I've been talking about here, there's an article that was just published in 2025 that indicated, remember I talked about herpes viruses in the brain? If you get trauma to the brain, you wind up getting Alzheimer's pathology, and this guy has shown that it's because trauma to the brain reactivates the virus infections in the brain. You know, instead of being latent, it reactivates them and stuff. Don't drink very much alcohol. Get exercise. Those things will reduce your risk of getting dementia. But that is really good advice. You don't have to be a rocket scientist. And no drug company is going to make any money off of it. But no, do it. You'll have a better quality of life anyway. Now, isn't anybody going to ask me about Prevagen? You see about it on TV? Well, unlike quality of life changes, Prevagen is bullshit. Okay? But quality of life changes are useful. Yes? Was that your question you were going to ask me?
Not at all.
Oh, good.
Just two-part question, actually. One is, you talk about it for Alzheimer's, MSS for Alzheimer's. It seems like, from what you're saying, but I want to verify, that it's for all dementia-related diseases.
It's not quite, I won't say all of them. I got another patent for the use of, excuse me. I got another patent, a second patent for the use of MSF for the treatment of cognitive problems after stroke. And that patent will expire in 2026. A lot of dementing illnesses involve acetylcholine. There are some dementing illnesses that don't. The dementia you get after syphilis and normal pressure hydrocephalus and tuberculosis of the brain, and those are probably not going to help.
And then second question is loaded. So as I've heard said, ain't nothing free. So what side effects do we have that are known from MSF?
The only side effects that we know about from MSF are that if you go more than 8 times the human clinical dose, if you go above that, you can get into trouble with nausea, vomiting, and diarrhea because it will eventually accumulate in the intestines. A more conservative answer is that we haven't had it in enough people long enough to know what may pop up. But right now, it's really clean. Yes?
Back to the Deprazole. Does that not cause vomiting?
Taking it in an effective dose. I'm taking it in the ineffective dose. The one that was used by the French people, the one that is barely above useless for dementia. But it's showed a good response. It seems to be more effective at slowing the atrophy of the brain. I would take a higher dose if I thought I could stand it, but. Are there any other questions? Yes?
Is it possible to diagnose Alzheimer's now, not the gene, but actual Alzheimer's in a living person? Repeat the question.
Okay. Is it possible to diagnose Alzheimer's disease in a living person right now? Not just the gene, but I said a lot of us in this room have it. And it's developing in our brains slowly. We haven't gotten to the point where we've got dementia, but it's in our brains and it's developing. The answer is yes, you can, but that's done only for research where you take a positron emitter, chemicals that tag the amyloid plaques and the neurofibrillary tangles, and then you do that. It's not available for routine clinical medicine. Yes?
So when someone says they got diagnosed with Alzheimer's, what's that mean?
Well, thank you. That's a really good question. First of all, I forgot to mention that if you're having memory problems and you think you have Alzheimer's or you might have Alzheimer's, the first thing that you should do is you should go to a neurologist and do it immediately, because you might have something else that's not Alzheimer's that's treatable. I mentioned normal pressure hydrocephalus, a brain tumor, an infection in the brain. All these other things are treatable, and go there immediately. And what the neurologist will do, or whoever you go to, a lot of physicians are knowledgeable about Alzheimer's besides neurologists, psychiatrists, people internists, people who deal with older people all the time will be very good about Alzheimer's, but neurologists are probably the best qualified. But they will run you through tests for everything else, and they will check your family history, and if you don't have anything else, they'll say you probably have Alzheimer's disease. I can tell you, you probably have Alzheimer's disease because 80% of the people that have dementia have Alzheimer's disease. Being able to hit accurately 80% of the time is pretty good. And I don't even have to know you. Am I too cynical? I probably am. I apologize. Any other questions? Yes.
Are there any other studies that you've heard of that were making any progress? Anything on the blood-brain barrier? Is there anything going on? I saw something on C-SPAN about it, that they were working on getting through the blood-brain barrier. Is there anything you've heard of?
Yes, I've heard of a lot of things. I can't think of them right now. There are a lot of drugs that are in development, and they're not ACHE inhibitors generally. They are drugs designed to attack beta amyloid in the brain or to stop the hyperphosphorylation of tau or a lot of other things. And there are a lot of different strategies in development. Some of them are in phase 1, phase 2, and phase 3. But I don't know of anything that I would consider a breakthrough. Yes?
I'm very cynical and very frustrated, and I get the answer that Alzheimer's is just one form of dementia, and that to me isn't an answer. I have a sister who is lost, and she has dementia. And so what's the difference between Alzheimer's seems to be getting popular, a lot of television time.
Yeah.
What about dementia?
The question is, what's the difference between dementia and Alzheimer's disease? And your frustration is well founded. If you have dementia, that means that you have a long-term, generally irreversible loss of cognitive abilities. And Alzheimer's makes up 75 or 80% of dementia. It also overlaps with other forms of dementia, one of which is multiple infarct dementia from little tiny strokes that you have. That's the second most common cause of dementia. And those overlap with some other ones. They're all called dementia. And yes, Alzheimer's is one type of dementia. And it'll make up most of dementia, but go to your neurologist and see if you have multiple infarct dementia. Go to your neurologist and see if you have pseudodementia of depression, or if you have dementia that's the result of vitamin B12 deficiency. Those are also dementias. But unless you get a workup, you don't know whether the dementia is Alzheimer's or not. In other words, dementia is an umbrella that covers a lot of different problems, and Alzheimer's is the big one. And it's not just because it's getting more popular, that's the wrong term, it's becoming more common. Yes, it is becoming more common because we're getting so much older, so many more people are getting older, and we're becoming more aware of it. We're not having an Alzheimer's epidemic. We're just seeing an epidemic that's been existing all the way along. Did that answer your question? Yes.
Not entirely.
Okay.
But, thank you. But my sister was only 72, and she's digressed, she's 73 now, she's digressed to these, the latest is she physically can get up out of a chair and sit down in a chair, but she doesn't. She just doesn't seem to comprehend. She's very congenial and go, like, come on, let's go sit down here. Okay, but she doesn't.
Well, your sister, being 72, is certainly at an age where she could have Alzheimer's disease. How long has she had it? 4 or 5 years?
COVID.
Oh, well, COVID. There are also post-COVID confusion syndromes, too. And Alzheimer's disease does not typically, although it does happen, but it does not typically interfere so much with motor behaviors. You can generally do anything you want to do. If you have something that begins to have motor problems where you cannot physically get up or sit down and that kind of thing, it might be something else. Including Parkinson's has a dementia syndrome, and you have motor problems. As you progress in Alzheimer's disease, and it spreads wider and wider in the different areas of the brain, when you get into the terminal decline, and unfortunately Alzheimer's is a fatal progressive disorder. When you get into that, then it begins to interfere with motor behavior, such as swallowing, and then you die unknowing, essentially brain dead, in the end, if you don't die from something else first. Luckily, or not luckily, most people don't get to that point, the bitter end of Alzheimer's. Yes.
Is Nepazil the brand name Aricept?
Yes, Donepezil is the brand name Aricept.
Okay, and then that's the one that gives you nausea, diarrhea, and vomiting?
All of them give you nausea, vomiting, and diarrhea. But if don't take enough to give you nausea, vomiting, and diarrhea. Donepezil comes in two doses, 5 milligrams and 10 milligrams, and if you're going to start Donepezil, you will be instructed to start on 5 milligrams. Take it at night before you go to sleep at night to help avoid the risk of nausea, vomiting, and diarrhea. Let your body get used to it for 3 weeks or so, and then try 10 milligrams. And if you don't get nausea, vomiting, and diarrhea on 10 milligrams, then stay on it. If you do, drop back to 5, stay on 5 for a few weeks, try it at 10, and they have been titrated. These drugs have been titrated so that 90% of the people can take these ineffective doses and not have nausea, vomiting, and diarrhea. Is that it?
Can I just say a moment?
Yes.
I'd love to end this on a little positive note. We're working with.
I thought this was positive.
Working with we formed a corporation on memory science tech, and what we're doing is we're organizing to take MSF through the clinical trial and get it approved. This thing is just set in the world.
Hold the mic up to your mouth.
I'm sorry. I'm so sorry. We formed a corporation, Memory
Closer.
Science Tech. Memory Science Tech.
There you go. Speak up, John.
I'm so sorry. We have a chance to get this approved by the FDA. We know the steps that need to be done. A huge amount of work has been done to get us here. We can do it. We can, at long last, bring something that works instead of doesn't work. We need some help, of course. I'm going to be in the second floor in the library tomorrow from 9 to noon time if anyone would like to understand the steps we have to take to get MSS approved. I promise you that we can help, all right? So please come in We're looking for your ideas on how to do it. We all need this, okay? Thank you.
That'll be in the river room tomorrow. Yes.
I wondered about the two drugs that are Kisula and Quimby, these two right up there, that are infusions. Do you know anything about those?
No, I don't. Lakimbi, and no, they're not the ones that attack the tangles. Those are lecanemab and adalimumab. It's some bab, meaning monoclonal antibody at the end. I don't know what those are. Now, some Alzheimer's drugs are not to treat dementia. They're to treat agitation and other symptoms like sleep, and I don't know about those.
Yes, one of these drugs, and I'm not sure which, is being, is in maybe a trial, a clinical trial, that my sister-in-law, she's 72, she's not very far along in Alzheimer's, and they recommended that maybe she could do one of these.
Okay, her sister-in-law is in a clinical trial for Alzheimer's with some drugs that I don't know about, and there are drugs in clinical trials that I don't know about, and they generally attack a different target and that kind of thing, and I just don't know about that. I'll tell you, you've been a wonderful audience. Thank you so much. Really appreciate it. And I've gone way over, but thank you.